Hello everyone, my name is Jeonga Kim and I'm a postdoctoral researcher at the KAIST AI.CRED Institute. Today I would like to present my research on the molecular dynamics guided computational design of lipid nanovesicle drug delivery systems. Before diving into the research, I would like to briefly explain the computational methods I utilize. Computational approaches operate across different scales based on different theoretical frameworks. Quantum mechanical simulations operate at electronic scales and are widely used to study chemical reaction mechanisms. Then there is molecular dynamics simulation, which provides atomistic level information. Beyond that, we can analyze macrodynamic behaviors and mechanical properties at larger spatial and temporal scales. My background is in material science, where I previously focused on understanding the electronic structures of the catalytic materials using quantum mechanics. However, because quantum calculations focus primarily on electrons, they face limitations when dealing with large systems containing numerous atoms. To address this, I have extended my research to MD simulations, which allow me to investigate the behavior of large systems, including biomolecules, over longer time scales. This is essential for the rational design of drug delivery systems. How does MD simulation work? We start by calculating the potential energy of the system to drive the forces acting on every atom. This energy includes bonded interactions and non-bonded interactions, such as van der Waals and electrostatic interactions. Using these forces, we integrate Newton's equations of motion to update the position of each atom, generating a trajectory. By repeating this process millions of times over femtosecond intervals, we can visualize the dynamic motions of molecules and precisely analyze atomistic mechanisms. Now, why focus on lipid-based platforms? While polymer and inorganic materials are common, lipid-based platforms are gaining significant attention due to their biocompatibility, high bioavailability, and tunable physicochemical properties. These advantages have made them a staple in FDA-approved nanomedicines. However, challenges such as low drug encapsulation efficiency still remain. To overcome this, I am developing multilamellar lipid nanovesicles to enhance stability and loading efficiency. By using MD-based design, we can overcome the time and cost limitations of traditional experimental approaches, which are often constrained by complex variables such as lipid composition, drug properties, interaction, stability, and physiological environments. Currently, I'm targeting Staphylococcus aureus infections. Staphylococcus aureus is a highly adaptable, opportunistic pathogen that evades the host immune system and causes systemic infections across various organ systems. It is one of the most lethal bacterial pathogens. Furthermore, its antibiotic resistance is a critical global issue. Despite extensive efforts, no vaccine candidate has successfully proven its efficacy in clinical trials. My research aims to develop multilamellar lipid nanovesicles-based delivery platforms to encapsulate antigen proteins for a more effective Staphylococcus aureus vaccine. Furthermore, I am modeling biological pathways, specifically endosomal escape, after cellular uptake via endocytosis. Nanovesicles are typically trapped inside endosomes. It is crucial for these particles to escape, otherwise they are degraded by lysosomal enzymes. I am using MD simulations to investigate whether mechanisms like membrane fusion or electrostatic interactions between the endosomal bilayer and the lipid nanovesicle can induce membrane instability, potentially allowing the protein cargo to escape into the cytoplasm. Beyond delivery, I'm also investigating the functional mechanisms of antigens once they are delivered into the cell. In general, antigens are degraded in vivo and exert their function by binding to MHC-I or MHC-II proteins. However, the detailed interaction mechanisms and key sequence determinants are still not fully understood. To address this, I first screen the potential peptide sequences using an immune epitope database. Then I predict peptide MHC binding complexes using AlphaFold 3 and subject the highest confidence models to MD simulations to evaluate their stability. This workflow demonstrates how the peptide remains stably bound within the binding group. This AI and MD integrated approach can be broadly applied to protein engineering, peptide-based therapeutics, and various drug design applications. Overall, my research focuses on three key directions: molecular dynamics-based antigen delivery using multilamellar lipid nanovesicles for Staphylococcus aureus vaccines, modeling intracellular delivery pathways, including endosomal escape, and combining AI with MD simulations to investigate antigen binding mechanisms. Looking forward, I plan to expand my scope to include diverse lipid species and functionalized molecules to build comprehensive lipid datasets. I also aim to extend these applications to various diseases and therapeutic agents, elucidating fundamental mechanisms like receptor binding dynamics. I'm open to collaborations in these areas, so please feel free to reach out. Thank you very much for your time and attention.